for the discussion on the full picture moonshot.
Thank you very much, Tata. So could we have the next slide? Yeah. So the moonshot, the full picture, I think if you compare with the other three, moonshot is the one that's most focused on mechanisms of disease by looking at the entire individual. And by that, I mean at immunological mechanisms that drive pathogenesis. Now, the most important part in that sentence is really the immunology. So still most of the immune-mediated diseases, as Tala explained, are really geared at looking at an organ, a diseased organ. Whereas here we really want to look at the immune driving part of the disease and how this is between individuals with different IMiDs. Now, where we can learn most is indicated on the next slide. Because as Tala also elucidated, much of the research within IMiDs is done in silos. So for each disease, a lot of work has been done, but there's not a lot of commonality across IMiDs. And this is what we would really want to change. So we know all of these diseases have clinical heterogeneity, and that likely reflects the complexity of a pathogenic immune response. And that part is still very much not understood. So indeed, this is more mechanistic and pathogenesis, and we've identified multiple levels at which we would like to address the immune mechanisms that lead to pathogenesis across IMiDs, and specifically address heterogeneity. So in the first bullet is one big problem. So for now, it's very often not considered what the role is of age, sex, and genetic ancestry. Sometimes it's done within a particular disease, but not across immune diseases. So it's very difficult because if you don't have enough power to really make conclusions on, for example, age and sex, but if we group all data together, we can much better learn whether differentiation in these types of parameters needs to be done to improve therapeutic strategies. So these are typically intrinsic factors, but we also have extrinsic factors like nutrition, microbiome, infections, vaccinations. All of these will also modulate disease and will come on top of that heterogeneity that comes from age, sex, and genetic ancestry. We also need to much better consider the entire patient. And by that, I mean the entire immune compartment. So often a lot of studies really focus on blood monitoring or on disease, but they don't ask what is the relationship between what we see in the diseased organ and what we see in the peripheral blood or in terms of systemic comorbidities. And these types of analysis are quite similar when you look across IMiDs, but have never been done in a disease overarching manner. So current IMiD therapies do not really take this compartmentalization into account. And you can imagine, for example, if you think of the immune system, most of the diseases are chronic. And chronic means that there is memory in the individual, memory that has been derailed and that should be treated. But these memory T cells and B cells are often stored in the diseased organ. So if we want to understand how to treat the immune response, we need to understand what type of immune response is occurring in the organ and how does it differ from what we see systemically. Now, last, a very common denominator that's seen across IMiDs is that at a certain point, because of the chronicity, patients develop irreversible tissue damage, and this can also be fibrosis. So this is a disease stage that's extremely hard to treat, and often this will involve some kind of more severe treatment strategy. Like, for example, if an intestine is damaged, likely it's going to be resected. So these types of very invasive treatments you would want to avoid. And so we really need to better understand at what immunological stage there's intervention possible and how could we prevent tissue damage and fibrosis better. Now, the value of cross-IMiD approach, I think I already stressed, is we're doing many of these things, but we're not making use of each other's data. So I think it's very important to see how can we extract these data from all IMiDs and what can we learn? And we have much more power if we combine all our data. And in the next slide, I would like to show you the approach. So in the Netherlands already, we have large, well-defined data sets, and we are collecting new patients in new cohorts. And if we do this together, we can much better get grip on the role of these intrinsic factors like age, sex, and genetic heritage, the roles of extrinsic factors like nutrition, microbiome, antibiotics, vaccination, but also how do responses relate within the individual. And here we would really like to spatially map and functionally analyze, for example, antigen specificity, memory versus not memory, in blood, lymph nodes, spleen, and bone marrow to get much better grip on how can we influence these responses. So, of course, the role of tissue remodeling and fibrosis is something that's also addressed quite a lot, but again, not in the commonality of IMiDs, and this is where we have a lot to gain. So on the next slide, this is the long-term objectives that we've defined, and what we would want in 2030. We would like to have a much better definition of these intrinsic factors like sex, age, etc., and understand which immune networks are really critically influenced by these intrinsic factors. In 2035, we would have to want to have much better grip on the spatially resolved immune landscape by putting all our data together and asking in what disease, at what time, or in which diseases together do we have particular relationships between blood and organs or lymph nodes. In 2040, we would want to have a better understanding of this last stage of most IMiDs where we have severe tissue damage and how we can get much better grip on that. So in the next slide, I have a few examples. So this is one of the examples of external factors that we've studied, and we've done that in TIMID, the TIMID project. What the problem was that we see changes in commensal intestinal microbiota across many different immune-mediated diseases. And it's often said that when there's this dysbiosis, that would fuel inflammation. But actually, the evidence for this is really slim. So what we did in TIMID is we asked across many immune-mediated diseases, do we see whether the immune system perceives changes? So what we did is for that, together with multiple groups within the Netherlands, we developed a high throughput assay to detect IgG antibody responses against intestinal microbiota. And we used taxonomic and functional profiling from fecal microbiome data that comes from a really big cohort in Groningen. And we typed what microbiota dysbiosis is common in different IMiDs. And from that, we deduced more than 70 strains that we cultured and that we then made lysates of. And then to these bacteria, we assessed whether there were IgG responses. And then we tested 874 IMiD patients. So this would be IBD, rheumatoid arthritis, celiac disease, psoriatic arthritis. We test all of these patients, also different age groups, 65 plus and pediatrics, all combined, and then asked, are there public IgG profiles? Do patients recognize microbiota? Do we see increases? And the answer is yes, we do. And we see public, so shared IgG profiles between IMiDs, but we also see very private IgG profiles. So responses to microbiota that only occur in one disease. So I think this is an example of how we learn. Yes, the answer to the question was yes. In immune-mediated diseases, changes in microbiota are perceived by the immune system, but we also can compare how different they are and how similar they are in immune-mediated diseases. And in the next slide, I would want to explain a bit better this idea of immune compartmentalization. I think most of us are probably familiar with the fact that when you study a response in a mouse model, you can study a response from time of initiation, you can study it until chronicity, and you can study it in all different organs. But in humans, when patients with an IMiD come in, they already have ongoing disease. So we cannot study it from time of from before diagnosis. And the other thing is we usually only have very limited tissue available, like blood and tissue. In our case, for example, if you do intestinal disease, a biopsy. And that makes understanding immune responses difficult because they differ in a different organ. In a tissue, as I said before, you'll have more memory. So then how do you understand whether what's happening in the blood reflects the tissue? Now, we've made a lot of progress in that by doing combined analysis in lymph nodes, blood, and tissue. And we're now progressing into that in this current project that's called Immune Health Seed, where we much better understand the relationship between what's happening in the blood and in the tissue in patients in bigger cohorts. And in the next slide, I would just want to emphasize, this is a study by another group that's published, but it just shows you here very convincingly that if you look at different organs, you see that there's different cells in these organs. Like, for example, in the bone marrow, you see that neutrophils, all the blue that you see are neutrophils. So that means that the relative contribution of particular cells in a particular tissue is not the same everywhere. So this really needs to be taken in account. If we want to change treatment strategies, we need to know not only what cells are we targeting and what process, but also how is this process happening in different organs. So I hope I've explained this a little bit enough, but if there's questions, I can do that in more detail. I'll now give the word to Marike, who is going to be giving a few other examples.
Thank you very much. Indeed, let me get some other examples about these cross-disease approaches, cross-IMiD approaches that we have taken. And one of the things is when we were working in Target 2B and Immune Health Seed, then of course the pandemic hit. And we are all totally sick of it. We don't want to hear about it. But I think in this way, it will show how it also, the cross-disease approach, gave us some important answers. And in this case, we look at B cell differentiation as B cells via germinal center reactions after antigen recognition. They give rise to memory B cells and antibody secreting cells. And most patients with one IMiD or another, they all have in common that they take immune suppressive drugs. And for instance, when you look in IBD and RA, TNF inhibitors are quite effective drugs for a subgroup of patients in IBD and RA. Obviously, when the SARS-CoV-2 virus came along, patients and physicians and researchers, we all had the same question. When you take immune suppressive drugs, and if you now see the virus, either live or whether you see it in a vaccine, are you going to mount antibodies against that virus or against that vaccine? Are these immune suppressive drugs interfering with your protective immunity that you need to generate? So we investigated that in a cohort of both IBD patients and RA patients. If you would push Tane, and again, and again. Yes, then you can see that we followed patients during vaccinations, and we looked into healthy controls, the AC, we looked into IBD controls not treated with immune suppressive drugs, and the green line people treated with immune suppressive drugs. And you can see that after 2 vaccinations, patients are a bit slower to induce good antibody responses against the virus, but in the end they do. They are very protective. That's not a problem. You see the line of protection in the picture. But what was striking, and actually what we did not expect, is that the antibodies in patients that are being treated with TNF inhibitors, the antibodies against SARS-CoV-2 decline quicker over time. When you look 6 months after the second vaccine, their level is definitely lower than in controls of IBD or healthy controls. And that has been reported in different groups all over the world in the meantime. Now, if we looked closer now, not only into the antibodies, but try to understand the common mechanism, what's going on, we could see that if we looked at the B cells that become activated against the SARS-CoV-2, against the spike protein, you can see that in the TNF-treated patients, either IBD or RA, doesn't matter, but in all of these patients, when you have TNF inhibitors, you have less specific SARS-CoV-2 B cells over time. And since we are B cell immunologists, we dove deep into that with a lot of markers and very involved panels. And then we dissected this B cell response into all the different subpopulations that you see below. And consequently, what we could see mechanistically, if you tap again, Tale, that you see that there is one specific cell type that is deleted or much less present when you have TNF inhibitors. And now the thing is that these cells are the precursors that we recently identified for antibody secreting cells, and they also contain precursors for IgG memory B cells. Summarizing, what we could see is that when you're treated with TNF inhibitors, your germinal center output, the reactions that give good B cell responses, is diminished. And for the inside people of us, the extrafollicular response was not affected. What I'm trying to show here is that only by means of our cross-disease consortia, we were able to investigate these questions and compare the role of TNF inhibitors both in IBD and RA, and could see that basically, although patients have very different organ involvement, their reaction to TNF inhibitors is the same, and also the effect of TNF inhibitor on generating protective immunity is the same. So that this research was nationwide research. The antibody data were from thousands of patients, was only possible because we united specifically all over the country with all the clinicians and could analyze many cohorts during vaccination. In the next slide, we took a bit more care of these IgG activated B cells by now knowing that they are precursors to the antibody secreting cells and the memory cells. And we also know that we only find them, for instance, in blood, shortly after you see an antigen. And we are now in ImmuneHealthXL exploring the following concepts. In many IMiDs, we know that we don't know the autoantigens against which autoreactive antibodies are formed. We don't know what the autoantigen is that the B cells recognize. That makes it very difficult to study immunologically, antigen specifically, what's going on in these patients and whether they react well to therapy. Now we know that these IgG-activated B cells only occur in blood relatively recently after antigen encounter. So we thought that patients with autoimmune disease that continuously see antigen, autoantigen, maybe their autoreactive B cells that have been recently and continuously activated are also present in this compartment specifically. Even if you don't know the autoantigen, what is happening with this compartment? If you tap again, Tana, you could see in a very small pilot study at the moment, we are hoping to extend it much more, that on the right-hand side in the circle, red circle, you see few cells in this IgG-activated B cell compartments. But in different diseases, you can see in this case myositis and myasthenia gravis, you can see that patients with autoantibodies have an enlarged compartment of the IgG-activated B cells. Now you understand that we're now doing big studies, isolation, Rhapsody studies, single-cell RNA-seq, and also trying to identify the B cell receptors, see whether there are clonal developments that might indicate the autoreactive B cell responses in this compartment. And this is what I wanted to show you as examples of how you can do overarching research and how that strengthens your data. Yeah, if you look at myositis or vasculitis, and if you have small diseases, it's difficult to study. But when you study the same phenomenon across IMiDs and you see common mechanistic pathways underlying it or common responses, you can dive deeper into that. Next slide. We have some